We're now going to be talking about Chapter 10, Muscle Tissue for Anatomy and Physiology, Bio 246, and we will start right off the bat with our objectives for the first section for Introduction to Skeletal Muscle. We must be able to explain the 5 general functions of skeletal muscle after repeating this section or going over this section, and describe the 5 characteristics of skeletal muscle tissue. So with the function of skeletal muscle, with body movement, basically that's one of the primary focus points or primary functions of skeletal muscle, is to be able to move bones, make facial expressions, speak, breathe, swallow, maintenance of posture, to stabilize joints, maintain body position. In general, stability is to maintain position or at least maintain structure or maintain the integrity of the area. Protection and support is to package internal organs and hold them in place. Regulating elimination of materials. Circular sphincters control the passage of material at the orifices. And heat production. It helps maintain our body temperature, maintaining the homeostasis at an ideal temperature for everyone. That being said, the characteristics of skeletal muscle tissue: excitability. It has the ability to respond to a stimulus by changing the electrical membrane potential, which we'll talk about shortly. Conductivity involves sending an electrical charge, not change, down the length of the cell membrane. Contractility is exhibited when the filaments slide past each other. Again, we'll talk about these here shortly. Enables the muscle to cause movement, or create movement, we should say, or produce movement, more so than cause. Extensibility, ability to be stretched, or the capacity to be able to be stretched and return to normal. Elasticity, the ability to return to original length following a lengthening or shortening, is more toward elasticity, versus extensibility. You can stretch it, but it comes back elastic. What did you learn? Were the 5 major functions of skeletal muscle? You should be able to state those and explain the skeletal muscle characteristics of contractility, extensibility, and elasticity, and then how do these differ? For section 2, the anatomy of skeletal muscle. You should identify and be able to describe the 3 connective tissue layers that are associated with a skeletal muscle. Describe the structure and function of a tendon and an aponeurosis. Explain the function of blood vessels and nerves serving a muscle. Explain how a skeletal muscle fiber becomes multinucleated. Describe the sarcolemma, the T-tubules, and sarcoplasmic reticulum, and the triad of a skeletal muscle fiber, and distinguish between thick and thin filaments. You should also be able to explain the organization of myofibrils, myofilaments, and sarcomeres, and list and describe the structures associated with energy production within skeletal muscle fibers. Define a motor unit and describe its distribution in a muscle and why it varies in size. Describe the 3 components of a neuromuscular junction, and describe a skeletal muscle fiber at rest. So we have quite a few objectives for this particular section because there's a lot here. Each skeletal muscle is an organ. Multiple types of tissues are working together. We have skeletal muscle fibers, connective tissue, blood vessels, and nerves, essentially, our primary ones here. Our muscle fibers are bundled within a fascicle. A whole muscle contains many fascicles, and a fascicle consists of many muscle fibers. A muscle fiber is a muscle cell. The connective tissue components. We have 3 concentric layers of wrapping around these muscles and the fibers and the different layers of muscle groups and whatnot to form the entire muscle itself. The epimysium is the dense, irregular connective tissue wrapping the whole muscle. Then going down, going deeper, going from superficial to deep here. Perimysium is the dense, irregular connective tissue wrapping the fascicle. It houses many blood vessels and nerves. And then we have endomysium, which is an areolar connective tissue wrapping individual fiber, and it has a delicate layer for electrical insulation, capillary support, and the binding of neighboring cells, the innermost layer. So we take a closer look here. We have the tendon attaching the muscle to the bone. It's really when you try to get the bone to move, the muscle will contract and shorten, but it actually pulls on the tendon. That tendon technically pulls on the bone. People like to say that the muscle pulls on a bone, and first of all, there's no pushing, it's only pulling. Your joint actions can be a push or a pull, but the actual muscular action is always a pull. Right? It's going to pull on the tendon is going to pull on the muscle, the bone. The deeper fascia here is that outer fascial layer. We have our skeletal muscle itself here. The epimysium, this is the epimysium essentially here, right here, going contiguously with this tendon here. And then we have showing artery here and veins, which would be going basically parallel with these fibers here, and nerves here. So you see the difference in the nerves and the arteries and the veins throughout. There's a fascicle right here. It's a fascicle group or a bundle of fibers here. And then we have a perimysium surrounding a fascicle. And then we have the endomysium surrounding individual fibers. Okay. As far as connective tissue components, going forward here, attachments of muscle to bone or skin to another muscle can be tendons or an aponeuroses, or aponeuroses being plural, aponeurosis being singular. A tendon is a cord-like structure of dense, regular connective tissue with muscle to bone, typically, pretty much in general. Aponeurosis is a thin, flattened sheet of dense, irregular tissue, and that can be tissue to tissue. Deep fascia is dense, irregular connective tissue superficial to the epimysium. It separates individual muscles and binds muscles with similar functions, so it's tissue to tissue here, whereas tendon being muscle to bone. In this case, muscle to other muscles, potentially, separating the muscles. Superficial fascia is areolar and adipose connective tissue superficial to the deep fascia. It separates muscles from the skin. Now, blood vessels and nerves. Skeletal muscle is vascularized. It has extensive blood vessels. It delivers oxygen and nutrients, then removes waste products and gases with the oxygenated blood. Arteries delivering oxygen and nutrients, and then the veins, you get blood moving away, moving the waste products and the gases. Skeletal muscles are innervated by somatic neurons, and these somatic neurons are axons of the neuron branch that terminate at the neuromuscular junctions, also known as the motor end plate, and that allows for voluntary control of contraction. For the muscle cell itself, muscle fiber or muscle cell, same thing. Sarcoplasm, cytoplasm essentially, has the typical organelles plus contractile proteins and other specializations. Has multiple nuclei. The individual cells are multinucleated. Cell is formed within an embryo when the multiple myoblasts fuse, and some nearby myoblasts then become undifferentiated satellite cells for support and repair of muscle fibers. The sarcolemma is a plasma membrane. It has T-tubules, also called basically transverse tubules, T-tubules for short, that extend deep into the cell. And the sarcolemma and its T-tubules have voltage-gated ion channels that allow for conduction of electrical signals. And it has a voltage-sensitive calcium channels also that are responsive to the electrical signals' action potentials, which we've learned before with cells in general. Taking a closer look at our skeletal muscle fiber here. So again, we have the skeletal muscle with epimysium around it, and the paramysium surrounding individual fascicle, and then individual fiber with endomysium. And around that, basically that single fiber here, or muscle cell, we have our sarcolemma, or plasma membrane here, and we have nucleus here. These openings in the T-tubules, the transverse tubules here, our myofibrils, or bundles of myofilaments. Okay, we have a nucleus here, our mitochondrion. And of course, we have our sarcoplasm on the outside of each of those. And take a closer look here, showing this myofibril. That myofibril here, this long cylindrical shaped muscle fibers in general are cylindrical shaped. Okay? And the more they will grow, more hypertrophy wise, will get bigger, but they also have that long cylindrical shape when they're utilized properly too. So we have our T-tubules here, each in between here, our terminal cisternae on either side here, our sarcoplasmic reticulum on the outside there, and each sarcomere is shown here, each individual sarcomere, this little separating lines, which we'll talk about here in a little bit. And then we have our myofilaments, our protein filaments here, which would be internal. And the triad, of course, referring to that T-tubule and terminal cisternae. Okay. For our sarcolemma and our T-tubules, we have our interstitial fluid on the outside of that muscle cell, right? We have our voltage-gated sodium channel, like we do with a lot of cells here, and our potassium, voltage-gated potassium channel. And of course, like most cells, sodium wants to enter and potassium wants to leave to help balance the equation on the both interstitial fluid and the internal aspects of the muscle fiber in this case here, past that sarcolemma, that membrane here, inside the cytosol, the cell. And of course, here's a T-tubule separating each individual muscle fiber. And the parts of the muscle cell, keeping going here, our myofibrils are hundreds to thousands per cell. There are hundreds to thousands per each cell of myofibrils in a given myofilament. And in our given muscle cell. Our bundles of our myofilaments are enclosed in a sarcoplasmic reticulum. The sarcoplasmic reticulum has an internal membrane complex that's similar to the smooth endoplasmic reticulum. Terminal cisternae, they're blind sacs of sarcoplasmic reticulum. They serve as reservoirs for calcium ions. There's two cisternae with the T-tubule in between. That's known as a triad, because there's two cisternae and then one T-tubule. Contains calcium pumps that import the calcium, and the calcium binds to calmodulin and calciquitrin calsequestrin, excuse me, and it contains calcium release channels. And these are triggered by electrical signals traveling down the T-tubule, and the calcium is then released into the sarcoplasm. So here's our triad here, our terminal cisternae. There's a T-tubule with the cytosol here and the terminal cisternae on the other side. And in between, of course, here between the cytosol, excuse me, and the T-tubule, we have the voltage-sensitive calcium channel here, and the calcium being released into this channel here, the cytosol. Here we have calmodulin here, and a calcium pump, calcium being pumped into from the cytosol into the terminal cisterna here. And of course, here's calsequestrin as well. They'll keep showing up a lot here. For the microscopic anatomy of our skeletal muscle, the myofilaments are contractile proteins, then myofibrils. There are two types of myofibrils, our thick and our thin filaments. Our thick filaments consist of these bundles of many myosin protein molecules, these myosin heads that point toward the ends of the filament, like golf club heads, shape-wise. Then we have our thin filaments. Consists of bundles of many myosin protein molecules that are twisted strands of F-actin, each F-actin composed of G-actin monomers. G-actin is myosin binding sites where myosin heads attach. Tropomyosin and troponin are present with regulatory proteins here. So we have here, so we have our muscle fiber, we have our myofibril and the myofilaments inside there, our myosin molecule. We have the heads, these golf club heads, these myosin heads, these actin, which binds with actin, the binding site for the actin to the myosin here. And these myosin heads here are protruding out from this thick filament. Then we have our thin filament. We're going to have our actin playing a role here. And we have, in this case, this little green strand, vine-like looking guys, our tropomyosin, our troponin here, our calcium binding site at the end of the troponin there. And F-actin is these guys between, and G-actin here. As far as the organization of a sarcomere, the myofilaments are arranged in repeating units called sarcomeres. They're composed of overlapping thick and thin filaments. Each of these is delineated at both ends by a Z-disc, and these specialized proteins are perpendicular to the myofilaments, and they are anchors for the thin filaments. The positions of the thick and thin filaments give rise to alternating I bands and A bands. As far as the I bands go in the sarcomere, they're the light appearing regions that contain only the thin filaments. They're bisected by the Z-disc. They get smaller when the muscle contracts and can disappear within a maximal contraction. An A band is a dark appearing region that contains thick filaments and overlapping thin filaments. It contains the H zone and the M line, and it makes up the central region of the sarcomere. The H zone is the central portion of the A band. Only the thick filaments are present, no thin filament overlap, disappears with maximal muscle contraction. The M line is the middle of the H zone. There's a protein meshwork structure here with the attachment to the site for thick filaments. So we take a look at this guy a little closer. There's our muscle fiber as a whole. There's each individual sarcomere here. Okay, and with each individual sarcomere, the separating is a Z-disc between separation of each sarcomere here, and the I band is basically the area where the Z-disc is in the middle of that I band here, and the A band is between here, and the H zone is in the middle, technically, completely in the middle of this sarcomere, between each of the Z-discs, and with the M line right here in the absolute separate middle here. So at M line middle, H zone houses that M line, and the A band is the greater area from there, and then it extends outward. Then you've got your I band with the Z disc contained inside of that. And then as far as the structure of a sarcomere, and you can bet at some point you'll probably have to identify that in a diagram, probably in a quiz at least, if not in an exam. For the longitudinal section of a sarcomere, we've got their entire sarcomere again with our Z discs on either side there. And again, we have our I band here, which separates across the Z-disc here, connectin here, with our thin filaments here, the actin strand there, and our myosin here, the thick filament, okay, and our M line separated between H zone again, basically being that middle point, middle area where the M line is housed, okay. And as far as a transverse or cross-sectional plane across the sarcomere, you can then separate and then see, here's the M line, the thick filaments and accessory proteins. Our H zone looks like this, our thick filaments. Our A band has got thick filaments and thin filaments. I band is thin filaments that are with connectin. And then our Z disc has thin filaments and connectin and accessory proteins. All other structural and functional proteins, our connectin extends from the Z disc to the M line. So it's like from the outside to the middle, essentially. And it stabilizes the thick filaments and has spring-like properties, providing passive tension. Dystrophin anchors some of the myofibrils to the sarcolemma proteins, and there are abnormalities of the protein that cause muscular dystrophy in some people. With muscular dystrophy, it's a collective term for hereditary diseases where skeletal muscles degenerate. And Duchenne muscular dystrophy, DMD, is the most common type. It's defective or insufficient dystrophin, and the sarcolemma is damaged during muscular contraction, and calcium enters the cells and damages the proteins. Problems begin in early childhood with this: walking difficulties, muscle atrophy, postural issues. It's incurable, unfortunately, and patients that rarely live beyond the age of 30. We're going to come back. I just want to double check my spacing. Yep, we're going to come back and finish talking about section 10.2b with microscopic anatomy of skeletal muscle and 10.2c here shortly.